Unit 9. And Unit 9, to be honest, the distribution of questions in Unit 9 is less in June and more in December net exam. Generally the question you'll get including B and C together can be 5 or 6 in June, where in December it may range from 8 to even 11, depends on year to year variation, but 8 to 10, 11 questions in December sometimes. Okay, now second part, what you need to prepare regarding Unit 8. Unit 9 is classification biology and like it's a biogeographical realm things. So this is where the classical zoology come in play, right? So as many students appear from the zoology background, you already have a understanding of the classification scheme in the biological classification system, and this scheme is for the animal classification system. Okay, and in this animal classification system we can divide Unit 9 into mainly 3 different sections. First section is those animal classification part. So from there you will get 3 questions including B and C. So if we only talk about C, 1 or 2 questions. Second part is cladogram analysis, 2 questions, and the third one is from the flora and natural flora and fauna of India and different parts of the India. Okay, so this is kind of a biodiversity link portions from India, 1 to 2 questions from there as well. So this concludes the total number of questions. Apart from that, there is one more thing, that is the concept of domains. They tend to ask questions from concept of domains earlier in NET, but recently in past 3 exams I haven't seen much questions coming from that part, that is archaea, bacteria and eukarya, that domains, and regarding that question. Okay, but cladography is a thing that is always given almost every year, either giving you the picture of the cladogram in the analysis or question in group C that helps you to understand the relation between different organisms, which is required. So almost 40% of the questions goes from this cladogram analysis, which is really important to do. Apart from that, from basic classification of both chordate as well as non-chordate, you need to read for the both, but for the non-chordate you need to just go from the phylum till phylum level of classification. In case of chordates you need to go till class level of classification. That's what you need to know, and obviously you need to know the hierarchy of classification in the first place. Okay, so from hierarchy even sometimes they ask one question in group C. So that concludes the total number of questions you can get from group C. It's ranging from 3 to 7 even. Okay, so it depends on the situation, less in June, more in December. Okay, now let's move on to unit number 9. Today we will talk about those animal classification scheme. We'll talk about the hierarchy of the animal classification system. Then we'll talk about the non-chordates and chordates, and non-chordates till phylum level, in chordates till class level. Now remember one more thing, this chapter, most of the part of this classification biology, that requires your memory skills to remember a lot of different features of organisms, okay, of different phylum. But in the part of cladogram analysis or phylogenetics, that is the part where exactly you need the explanation, you need the understanding. So my job is there more to help you understand cladogram analysis and solve few practice problems so that you become prepared for the type of question you are going to get in the exam. But mostly unit 9 is like approximately 80% memory based, 80 to 90% memory based. Even in the cladogram questions, they ask questions where you need to input some of the knowledge that or some of the information which you need to remember for a long time. So it's better you prepare with the blueprint of life sciences for this part. Along with it, I'm just going to tell you a few important portions where you need to focus more while you're preparing this. And one more thing is that while you're preparing for the different phylum classification, I've recently posted a mnemonics video on that. I hope you've seen that video, where you can easily remember how exactly you can understand and construct that that classification scheme till phylum level in case of non-chordates. Okay, so let's talk about the classification scheme hierarchy in the first place. The classification scheme hierarchy is pretty simple and pretty easy to remember. Dumb King Philip come over from German soil. It's very easy to remember with this mnemonic, where it goes with dumb, D for domain, K for kingdom, P for phylum, Philip, then come, C for class, over, O for order, from, F for family, and German, G for genus, S soil, S for species. So that's how the hierarchy is organized: domain, kingdom, phylum, class, order, family, genus, and species, where the domain is the largest and the species is the smallest. So when we talk about a genus contains many species, many organism species together is a genus. When we talk about a family, it means many genus together forms a family, many families together order, many orders together a class, many classes together a phylum, and many phylum together a kingdom. That's how they are all arranged. Now, broad classification of animal kingdom. How exactly we will do the classification in the broad scale? Animal kingdom can be divided into unicellular animals and multicellular animals. Unicellular animals, example protozoa, they are actually known as protozoa. Multicellular animals known as metazoa. Now the metazoa can be further divided into invertebrates and vertebrates, where invertebrates, we need to read invertebrates till phylum level, vertebrates till class level. We'll talk about that. So when we talk about vertebrates listed, fish, amphibian, reptiles, birds, mammals, they are all the classes of vertebrates, where the invertebrates, we are looking sponges, coelomates, worms, arthropods, they are all the phylum that are classified based on their properties. Now, a very simple question that you can ask yourself, that how exactly we classify organisms these days. Okay, earlier the classification of organism was purely based on the morphology, its structure of the organism. So by looking it from outside, we can say which organism is similar to which organism. But now we mostly focus on the genetic understanding, that is the genetic similarity and dissimilarity between the organisms. We also focus more on the components that are common between them, in genetic component mostly, and the biochemistry of that organism, the physiology of that organism, and many more things correlated together to finally comment on thing. Because if you comment based on the physical appearance of that organism, most of the time you will make mistake. For example, if we always measure that physical appearances, then we can never think of whales or dolphins as mammals, because whales and dolphins are much similar in terms of physical appearance with fish like shark, but they are totally different. They are different in terms of many different aspects. Now we know that because we did not limit our understanding based on the physical appearance. We always try to think of the genetic changes and modification, and the same thing, this genetic modification and physiological modifications, as well as the anatomical changes and interactions, or everything we need to take account for, because all these values, all these informations not only help us to identify and classify organisms, but also help us to identify how they are originated in evolutionary history, how exactly they are originated, from where they are originated, what is their ancestor, and whether that ancestor has that similarity or not, whether they are derived with new characteristics from their ancestor or not. We can get this idea all along. Okay, so it's not only just to classify the organisms, but also it's very, very important to find out the relation between the organisms in the evolutionary time scale as well. So now let's talk about the characteristics of classification. Now, classification means we are classifying organisms based on what? Based on characteristics, like as I said earlier, the appearance, obviously, then morphology, physiology, anatomy, everything is a part of the characteristics, and based on these characteristics we classify the organisms, right? So the characteristics of classification is based on in several different levels. With different levels we divide them, because we have one big kingdom here we are talking about, that is Animalia. Because three big kingdoms, if we talk about the five kingdom concept, if you remember, the five kingdom concept, although it's not the most modern theory, the most modern theory is the three domain, where we only focus on very fine differences in their genes between eukarya, archaea, as well as the prokaryotes or bacteria. Now in this case we're looking at the kingdom in a very simplistic manner. Two separate kingdoms we can easily mark if we look at an organism. That can be either a plant or an animal. So we can divide them into an animal kingdom or a plant kingdom. Fine. Now if we're looking at the animal kingdom can be of two different types. Remember, as I said earlier, it can be protozoa, it can be metazoa, like multicell or a single cell. A single cell organisms are totally different group, okay, and a multi-cell organism a different group. But multi-cell organism means that not only means they have multiple cells. Now the question is how exactly those cells are interacting with one with another, whether they remain at the cellular level in terms of their function, or they form tissue, or they form an organ system. Now depending upon that, we can also divide that animal multicellular kingdom. Okay, so the first property for which we can divide is the levels of organization. And levels of organization means either cellular level or the tissue organ system level. So cellular level is much basic, tissue organ system level is little bit complex than the cellular level. So cellular level can there is only one type of phylum belongs to the cellular level. Example, Porifera. Example, sponge, belongs to cellular level organization in the animal kingdom. So Porifera phylum organisms like sponge, they are multicellular first of all, but those cells that are present, they never interact to form any tissue or organ system. So they just presence in a scattered way. So if we put that cell, if we take the cell from the sponge and if we put it outside, that cell can originate another sponge, and that thing can be done again and again. That thing can be done. So that's the beauty of the cellular level organization of a phylum. If you take the one cell out of it, that cell can give rise to an adult organism. Now the rest are the tissue organ and organ system level of organization, second one, and that can divide into two different types, and depending upon the second important property, that is symmetry, body symmetry. Now what kind of symmetry we are talking about? The body symmetry we are talking about can be either radial or bilateral, either of this, radial or bilateral symmetry. Now the radial symmetry means you don't have bilateral symmetry is very easy to understand. If we slice it through the center axis, it will produce a mirror image of that organism with one another from the left side with the right side, while the radial symmetry is not that thing. If we separate them from the middle, the structures will be different in both the sides. So few of the phylum, they belong to this radial symmetry of the body, few and rest of the others are bilateral. Most of them are of bilateral. Bilateral symmetry came much later in the time of evolutionary scale, so it is thought to be a little advanced over the radial symmetry. The example of radial symmetry, Cnidaria and Ctenophora. Okay, that's a part of the radial symmetry. One more example of the radial symmetry, if you look at Echinodermata, they can also belong to that symmetry, but we put it in the bilateral because there is a reason. You know, there are organisms where they belong to a larva form, then an adult form. The larva form may belong to a different symmetry, while the adult form may belong to a different symmetry. Echinodermata in the adult form is a part of the radial symmetry. They do not have any bilateral symmetry, but bilateral symmetry is present during their bilateral symmetry is present during their larva state here. Okay, that's why we put it in there. Okay, so Cnidaria and Ctenophora phylum belongs to the radial symmetry. Now goes to the bilateral symmetry. Bilateral symmetry rests all. Now belonging to all the phylum with the bilateral symmetry. Now the third important property based on which we will separate them is the presence of body cavity or coelom. Now if I ask a question, what is coelom, what is body cavity, you remember one term, primitive gut, right? Primitive gut was formed at the end of the gastrulation process. We all know that. We have seen that for sea urchin, we have seen that for xenopus. So there this formation of this coelom or body cavity at the end of the gastrulation process, and that is why there are few organisms where the body cavity or the coelom is not formed at all. There are organisms where we have formed a body cavity, but that body cavity is not fully formed or fully functional. We call them false coelom or pseudo coelom. And there are organisms where the body cavity is formed and fully functional, because what's the role of this cavity? The intestinal organs, the internal organs live inside the body cavity. So there are organisms where the organs and systems, they are floating in their body without any kind of cavity, but here they produce a dedicated structure or body cavity where all the organ system is placed. Okay, so based on which we can say the acoelomates or without the body cavity, pseudocoelomates or false body cavity, and coelomate with true body cavity. Body cavity is also known as the coelom, right? So without body cavity, example platyhelminths. False coelom or pseudocoelom, example aschelminths. And the true coelom, rest of the other phylum. And what are the examples? Let me slide it a little bit here. Okay, the examples that we can see here, that annelids, arthropods, mollusks, echinoderms, hemichordata, urochordata, all of them, they belong to this true coelom organism. Okay, true coelom phylum. So this in a sense is the best picture that will explain all the phylum, and based on their different properties, how exactly we characterize them. So remember these three properties: levels of organizations, body symmetry, and body cavity or coelom, presence of coelom, that help us classifying all the non-chordates or invertebrates according to their phylums. So now at the end what we get is multiple phylums like Porifera, Cnidaria, Ctenophora, Platyhelminths, Aschelminths, annelids, arthropods, mollusks, echinoderms, hemichordates, and urochordata at the end, where we will see the formation of the backbone structure, which is the typical feature or unique feature of the chordata, which we'll see for the vertebrates. They form the vertebrae in the back, and then we will see the different classes of the vertebrae in the next part. So remember, this is the portion where they can ask a question. They can ask a question from the general hierarchy of the classification. They can ask you a question regarding this levels of organization or different properties based on which we classify organisms, or they can ask question from individual phylum regarding their properties. Now the question is, this picture not only explains the few important properties, because you can easily see the few important properties from this picture pretty clearly, but apart from that, this picture also states the role that there are important feature in the evolutionary role, how exactly organisms are produced. So Porifera at the very preliminary level of organism with only cellular levels of organization, then at the end chordata at the end, which produces with true coelom, right? So from Porifera goes towards chordata, and we know chordata at the end is much advanced in terms of body organization and structure. So this is a very important thing. So if you are remembering and try to recall the features, the characteristics of different phylum, then you should obviously look for this slide and remember based on this property based approach to remember. Otherwise it will be very difficult if you just mug up the features. Now it's time to talk about all these different phylum and their important properties. Now this part is solely a thing to remember, solely a thing to read and memorize. In this case you can form like for every single organism's few important properties which are unique, although because you know every single cases we know there is a similarity between Porifera and Cnidaria. The similarity is based on they are all multicellular. Now between Cnidaria and Ctenophora, the similarity is that they all share a tissue or organ system. Between Ctenophora and Platyhelminths, the similarity is that they all have again tissue organ system. Between Platyhelminths and Aschelminths, the similarity is that they both share bilateral symmetry. So you need to remember all these facts so that they can ask questions from the relatedness between organisms. They can tell you based on providing you the name of the phylum, or most of the time just providing you the name of the organism, and even to make it a little complex they can provide you the scientific name of the organism and then asking that kind of questions. So invertebrates, first of all, Porifera. Porifera has cellular organization, okay, and they are filter feeder. They are sessile, means they are attached, doesn't move around. The reproduction mechanism is asexual, fragmentation, budding, and sexual. And they contain two germ layers, endoderm and ectoderm. They do not have any kind of mesoderm in it. And they have the typical feature of pores through which they can take water filled with nutrients in and out, because as they are attached they cannot move. So the water transport through these pores are really important, and the water transport that works there is with osculum and ostia, right? So small pores through which the water moves, and osculum are the large pores. And they also have this spongy body known as spicule. Okay, their body, the outside structure is mostly made up with deposits from glass or silica, and a protein known as spongin. Okay, and calcium carbonate deposits that are present. Now the thing is, as they are their cellular level of organization, you take their cell, and if you just take a sponge and destroy it, break it down and throw it out from that cell, new sponge can form. Okay, the symmetry of sponge can be radial or asymmetrical. Cnidaria. Characteristics for Cnidaria: radial symmetry with true tissues. Now Cnidaria is a little advanced from Porifera, because for Cnidaria the cellular organization is converted to tissue. Okay, so they have formed the tissue layers. That's why if you look at the structure, example Hydra, example jellyfish. So if you look at here, Hydra, very tiny organisms here. Jellyfish is not that tiny though, but Hydra is very tiny, where they have mouth, they have tentacles to capture the food, and the cavity that is present in the center. Although the cavity is open, nothing is fragmented there inside, but a layer of tissue start forming outside, and that layer of tissue and surrounding it jelly like layer is present on the outside. So two separate layers, inner layer and outer layer, and jelly like layer is present in between the inner and outer layer. Okay, and another so the symmetry here is radial, because if we divide them from the middle, we don't have mirror image structures of each other, different. Now they have stinging cells known as nematocysts that help capturing the food. Now these stings can be fatal to humans. Example Hydra. Okay, as you see. Otherwise jellyfish, on the other hand, nematocysts are there. The stinging cells, the stinging structures are composed of special cells known as cnidocytes. Okay. Now there is an economic importance for this type of organism. The reason mostly because it affects human in many ways. Generally, to control the plankton population underwater is very important, because plankton population regulates the higher level of organisms in the aquatic environment or aquatic ecosystem, because the phytoplanktons are the one depending on which all the other organisms actually depend for the survival. That's one very important thing. Apart from that, humans like this, like jellyfish, because they store it as an aesthetic purpose for different underwater aquariums and stuff like that. Okay, and the major characteristics again listed here are radially symmetrical, apical organs, and they have this hermaphrodite sexually or asexual reproduction process. So till that last part, till sponge, there are three different ways, asexual, sexual, as well as the budding process was there. Budding process is also here in case of Hydra, but in this case there is another different characteristic, that is hermaphrodite. Hermaphrodite means an organism which can produce egg and sperm both, so behave like both male as well as female. They can involve in doing or producing both the type of gametes for the fertilization event. Now these organisms, they share characteristics like they have a medusa and polyp stage for their development. They are mostly marine, and they live in protective shell until they hatch, and the diffusion of oxygen through the tissues from outside. So these are the major characteristics. Let's move on to the third type here. Platyhelminths. Platyhelminths characteristics, organs and systems are present. Example of platyhelminths, first of all, tapeworm, right? Organs and systems are present. Bilateral symmetry, gastrovascular cavity is there, closed digestive system, but the excretory system is not fully formed. Protonephridia is present. You know, the highest form of the excretory system that is present is the kidney, and the unit of the kidney is nephron. But before that it was nephridia, and even before that it was protonephridia. So platyhelminths have protonephridia. Reproductive system, generally hermaphrodites are found. Nervous system, the most advanced form neurons, but here ganglia. Okay, that's the difference. But the problem is they don't have any kind of body coelom. Whereas helminths characteristics, first animal to possess a body cavity. Although they have a body cavity, but it is not covered, so it is not a proper coelom. It's known as the pseudocoelom. Distinct body cavity with no peritoneal lining, remember, and visceral organs lie free in that cavity. That's the difference. Another interesting fact about the aschelminths is that rest of the things are fine. They have the organ systems, everything is there, protonephridia, excretory organ. Reproductive system is little modified because still platyhelminths it was hermaphrodites. But in case of aschelminths, two separate gender distinction has been done. That is, male and female both are present. Male has the structure at the end which is little curved. One part of their body is little curved, while in female it is straight in both the ends, and females are longer, males are shorter in size. Annelids. Annelid characteristic, bilateral symmetry, body metamerically segmented, means segmentation from outside, and bilateral symmetry because if you divide them from center you have same structures in both the ends. And their body is chitinous outside. The digestive system is again complete. Respiratory system is gas exchange through skin, gills or parapodia. The circulatory system is closed. The excretory system a pair of nephridia. Now remember, we are going towards advanced structures. In the earlier case we saw the example of excretory system is protonephridia, but in case of annelids it is nephridia. Example of annelids, what? Hirudinaria. And also you can see here the very common example, what? Worms. Leech is also there. Mollusk. Mollusca or mollusks, they mostly found in seas but may be also found in fresh water. Their body is not segmented like annelids or arthropods. They show bilateral symmetry. Okay, they have an open circulatory system. Now this is a downgrade value of the circulation, because earlier than that the circulatory system was found intact, but now it is open circulatory system. It possesses kidney-like organ. So now the nephridia is being modified to form a kidney-like organ for the excretion. Their body is made up of 3 germ layers, so they are triploblastic in nature. But before that, those organisms, most of them, they are the diploblastic because they have only outer layer and inner layer. But here mollusks are triploblastic. And also from aschelminths you can find that layer differences, because these are all they carry proper coelom, okay, or true coelom organisms. They have a distinct foot for moving out. Echinoderms characteristics. Echinoderms have spiny skin because they have an exoskeleton, and the muscles are connected to it from outside. You can see that skeleton from outside. It's kind of unique, and we call it musculoskeletal system. And their body is 5-part radial symmetry, or known as pentaradial symmetry. They also have a organism. They also have a separate structure to move, tube fit. Okay, and they are benthic organisms because they are present at the benthic zone of the ocean. What was the benthic zone? Anyone? Lower, exactly. They are found to the lowest ranges and deepest portions of the ocean. Now another thing is the body temperature is ectothermic in nature. symmetry radial. As I said, pentaradial as an adult organism, but as their early growing or larva form, they were bilateral. Digestive system complete, skeletal system exoskeleton, circulatory system still open, reproductive system sexual asexual, heart chambers, no true heart is observed for this organism. Okay, now move to the next one. Arthropods. Arthropods are little different, that's why I put them here. Arthropods are different because arthropods are the most abundant phylum among all. They have lots of legs and segmented bodies. There are 4 group of arthropods that are present: arachnids, centipedes and millipedes, crustaceans and insects. All the 4 that should be present. Okay, so the characteristics of arthropods: 2 out of 3 organisms are arthropods in the whole world, so it's very much dominant. Segmented body, they have 3 body segments generally: head, thorax and abdomen. Few segments like cephalothorax, although the spelling is wrong, but cephalothorax, sometimes few segments are also found where head and thorax are combined together. Okay, open circulatory system, they use hemolymph and hemocoels as the circulatory system where blood opens up in their body cavity. Exoskeleton, just like echinoderms, they have exoskeleton system outside that prevents water loss and that helps in attachment of the muscle from inside. Now if we talk about all this, for example, arachnid is a totally different types of arthropods and particularly applicable for the spiders. They have 4 pairs of legs instead of 3. Generally when we say arthropods, we always mean insects, but insects are actually one type of arthropod, but there are different types of arthropods available. Like arachnids, they have 4 pairs of legs. They have bodies divided into 2 sections only: cephalothorax and the abdomen, because thorax and head is linked with one another. Okay, that's the difference, but all their legs are attached to the cephalothorax region. The same thing for the insects, all the legs are attached to the thorax part, not from the abdomen. And centipedes and millipedes, these are also another group of arthropods. They have long thin bodies and pair of legs on each of their many body sections. Okay, now here what we can see is that every single body segment, if you look at this way, every single body segment a pair of leg is there, and that number of legs are huge number of legs are present. That's why we call them centi and millipedes. But their legs that comes out from their body segment are also kind of similar in length from both the sides. This is unique because their body segmentation is more, not 2 or 3. They're totally different. Third type of arthropods are crustaceans. Crustaceans, they have 5-7 pairs of legs. First pair often used as pincers, as the structure to get the prey and break it down and to eat it. Body is covered in shell, and sometimes even though it's not covered in shell, but they find new shell to live there. Okay, example of crustaceans, you can clearly see scorpion as well as the other structures like that, and also marine, but not necessarily marine. They tend to live in the near seashore environments, few of them, and rest in the deserts.